Be very tiny, like a bacteriophage. As you suspect, a virus that affects very tiny cells, like a prokaryotic cell, is going to be very tiny itself. And that's what, I can use the mouse, there we go. And that's what this is showing right here. So this little alien spaceship is a bacteriophage. It is landing on the surface of a bacterial cell. So this is the envelope right here, the outside of the bacteria. And if you extrapolated that out, you can imagine how big that cell would be on this slide. So that's how tiny bacteriophages are. Protein capsid, made up of what we call capsid subunits surrounding genetic material. Basics for a virus. But we can have a couple of other additional elements on there as well. You can have some proteins or some glycoprotein structures, which we call spikes, extending out from that capsid. We can also have a lipid membrane that surrounds that capsid, called an envelope. So as you notice, points 3 and 4, not all viruses have those. But all viruses will have capsids composed of capsomeres surrounding genetic material, DNA or RNA, never both. So let's classify them based upon these structures. First off, your capsid shape. We're going to have 3 categories for capsid shape: helical, icosahedral, or polyhedral, and complex. So helical is more cylindrical, rod-shaped. Cylindrical or rod-shaped. The genetic material just kind of fits in it, coils. There we go. Could be as basic as like the tobacco mosaic virus, the first virus we ever discovered. Who cared about why our tobacco leaves were wilting back in the late 1800s? This can be unenveloped, like in the case of TMV, or it could have an envelope, like in the case of Ebola. Okay. Next, we have polyhedral, and the most common form is icosahedral. So icosahedral means that you have 12 vertices and 20 face sides. Who's my D&D nerds in here? Just me? Cool. There we go. Hey, we need to hang out more. Okay. What do we call that in D&D or any other board games where you roll a dice? D20?
D20, yeah.
D20, right? I mean, heard of D20? That's what a D20 is. If you don't know what D20 is, look up it. It's basically what these look like. For those of you that's more familiar, maybe more like a soccer ball. Rhinovirus is a good example of that. And then lastly, everything else gets thrown into complex. Well, if it's not a rod, it's not a soccer ball, then we're going to call it complex. And this can include a variety of different shapes. Some things look like a brick, right? Some can be a combination of both helical and icosahedral, and then we would still call that complex. For instance, like bacteriophages, they take on that look. They have an icosahedral head, and then they have a helical sheath. They work a little bit different. Okay, they'll store the genetic material in that icosahedral head, and then the sheath acts more like a hypodermic needle for whenever it's ready to insert the genetic material into the host. Okay, it'll have a base plate which tail fibers or tail pins will attach to, and then those pins act like little landing gear, I guess, for that host, and they're very specific on how these tail fibers will attach to the outer envelope of that cell, in this case, a bacterial cell. We're going to group them by whether or not they have an envelope. They have an envelope. Yes, they're enveloped. If not, we call them naked. And this is going to affect a couple of things about the characteristics of the virus. It's going to affect how it enters into that cell. And it's also going to affect how we treat these potential viruses. I'll give a great example in chapter 13 for this. We also categorize them by their spikes. So they have these protein spikes that come off of that capsid, and they aid in the attachment and the release from that cell. We categorize these spikes as hemagglutinin or neuraminidase spikes. So H for short or N for short. H spikes, N spikes. Have y'all heard of these before? You have, but you don't realize it. Y'all were alive in 2009. What was the flu scare that was going on then?
I was 8.
Ebola was, yeah, flu scare. Ebola was 2014. I can talk about Ebola today if you want. It wasn't? What kind? Swine flu, good. Swine flu, do you remember what they called it? Like H2N3, or maybe you've heard of H1N1. Guess what they're talking about? Oh, I told you already knew about it. Yeah, we categorize these viruses all the time by their spikes. So as you'll come to see when we start creating vaccines for stuff like the flu seasonally, we will look at these different archetypes of virus spikes, and they'll make a cocktail of a lot of different hemagglutinin and neuraminidase spikes within that flu vaccine. And we hope that we get it right so that you don't get the flu. Okay. Now we're going to group it by its genetic material. So there's, let's see, it shows 4 on here, but we're going to technically expand that a little bit more into 5 or 6, the way you want to categorize it, because once we get to single-stranded RNA, we say it's the positive or the negative sense RNA, or sense or antisense RNA. So single-stranded, double-stranded DNA, single-stranded, double-stranded RNA. Within single-stranded, you'll have positive and negative. I'll get back to that as we talk about the way that viruses replicate. So keep that in mind. Now, we don't include this in our big phylogenetic tree of life, the 3 domains, because we are acellular here. But that doesn't mean that we're not OCD about classifying things as scientists. So yes, we still classify it. So the ICTV, the International Committee on the Taxonomy of Viruses, maintains a database of viruses. And this is nonetheless important for medicine when we move on, because we will attach different types of alphanumeric codes to these viruses and follow it along and stuff. But because viruses have a unique morphology and they mutate pretty quickly, it's hard to put a species and a strain on a virus, and because they change up quite frequently. So instead of using the genus species epithet for typical binomial nomenclature, which we do for living, instead we use the family genus epithet. Right, so anytime you see Viridae at the end of it, that's going to designate a family. If you see virus at the end of it, it's going to tell a genus. So you think coronavirus, right? That's the genus of it. Coronaviridae would be the family of it. Just go ahead and cross this out. This is super outdated. They have like higher levels of taxonomy over there. They have like realms. They have like 200 orders and thousands of families. That needs to be updated, so don't worry about that. Just know that we know a lot more viruses now because we're a lot better at virology. All right. So we follow these viruses as unique as we can, and we classify what's known as an ICD code for them. So you'll recognize these. It's an alphanumeric code once you get further in your nursing career, and it's going to be unique to that type of viral infection. You're going to use this ICD code all of the time, or you may use this ICD code depending on what you're doing there. But say a patient comes in and they're showing signs of the flu. Well, you're going to assign diagnostic tests for that flu, and you have to use an ICD code for that. Once you diagnose it as the flu, then when you prescribe some type of treatment, say, all right, I'm going to give you some Tamiflu. There you go. You're going to attach an ICD code for that. Say they need to get reimbursed by their health insurance for all of that. Yeah, the ICD code is attached to that too. Say that they die from it. The ICD code goes on the death certificate. And then say we want to look at stats about how many people died from viruses that year. We're going to look at those ICD codes through epidemiological studies. ICD codes are pretty much used for everything in the whole process, going from diagnosing, to treatment, to healthcare, to insurance, to death, studies of death and prevalence and so forth. ICD codes are used for everything. Okay, let's go to life cycles. Once again, goal, take over the host and make your babies. It's going to be different if it's bacteriophage versus if it's eukaryotic. Let's do bacteriophage first because it is simpler and it's easier to do these studies. Sometimes it's more ethically apt to do these studies because we don't care about killing millions of microbes, and sometimes we feel bad about killing rats and bunnies. We have 2 different types of phages, and they have 2 different types of cycles. You have virulent phages, which go through the lytic cycle, and you have temperate, sometimes we call them latent phages, that go through a lysogenic cycle. So virulent phages will infect their host and then kill it by lysing that cell. Temperate phages will enter their host and then kind of hang out and co-replicate within that host cell until it's ready to enter into the virulent cycle, or to the lytic cycle, and become a virulent phage and kill the host. So let's look at the lytic cycle of virulent phages first. Know these steps in order. Number one, you have attachment with those tail pins and fibers to the outside of the host. Once again, very specific in the type of cell that they attach to. After they attach, then they will use that sheath to inject their genetic material like a hypodermic needle. So you can see it's putting its genetic material in there. That's blue. The host chromosome is pink. Okay. After they enter in, they're going to hijack the cell. So they'll express a number of proteins that allow them to chop up the host DNA, steal that machinery, and replicate their own genome. And so the polymerases are stuff that's like expressed first, and then later on you'll have more like lysis type proteins to kill that host. Step 4, maturation. They assemble their progeny. The virions are getting built. Now all look like little bacteriophages now. Last step, lysis. Bust open the cell, so you go from one to many. Lysogenic cycle, same idea, because lysogenic phages will turn into virulent phages. But first they go through the process of lysogeny. So we start out with the first two steps being the same. I'm not tall enough. So attachment and penetration. Right, this is right here in this first bubble. Attachment and penetration, see that occurring. Same thing, familiar. But instead what happens, directly going and expressing all of its virulent genes, this piece of genetic material will incorporate itself into the host. So you can see the pink host chromosome with a little bit of blue in there. At this point, this little blue part becomes what's known as a prophage. The whole cell itself is termed a lysogen. So the prophage is within a lysogen now. This process is called lysogeny. And it's going to hang out into there, and it's going to continue to replicate with the host. So whenever the host goes through binary fission and it replicates its own chromosome, it just hangs out and it's like, okay, I'm getting freely replicated here. And it goes into all of the progeny. Can spread. And then depending on some type of environmental cue, maybe a change in pH, maybe a drop in food supply, what's going to happen is induction. This is where the prophage will excise itself. Excise meaning it's going to cut itself out of that host chromosome, and then it's going to enter into the lytic cycle, become a virulent phage. So after it is induced, then it goes to what's step 3 of the last lytic phage?
Biosynthesis.
So we had attachment, penetration, now we're at biosynthesis, which is kind of a different word for replication. No, it's kind of the same idea, right? Biosynthesis, maturation, lysis. Good, so you can see that lysogeny is a part of the lytic cycle in this case. Does that make sense to everybody? It's still going to go through. Now, lysogeny contributes a few things to bacteria. As they add themselves in as a prophage, they can actually bring along additional genes just because of the way that they have evolved. In this case, it can contribute to virulence in a host. And this has been demonstrated in cholera and botulism, because those types of cells without their respective prophage are less virulent. And we've come to find that these prophages will bring in specific, more toxic genes or more virulent genes that affect the host in worse cases. Now let's go to the concept of transduction, meaning that this provides some genetic variability to host cells because of bringing in extra genes, just like I showed with botulism and cholera. We have two different types of transduction. Each one respectively occurs in whether it's in the lytic cycle or the lysogenic cycle. So generalized transduction, DNA is randomly capsuled up and brought to different cells through the lytic cycle. In specialized transduction, it's a little bit more directed. Let's look at how this occurs in both of them. So this is specialized. Let me take a step back here to the lytic cycle, and I'll show you what transduction occurs. So as the host gets into the cell and it starts expressing these genes and proteins to chop up the host's chromosome into little pieces so that the host can't really do anything, it's not like that DNA disappears. Those bits and pieces of DNA are still floating around in the host cell. And so whenever biosynthesis and maturation occur and it is encapsulating its own genetic material within the capsid, it accidentally sometimes takes some of the host's chopped up material. So there's bits and pieces of the old host within that capsid. It releases, and then it goes to the next cell, and it does the same thing. But this time when it injects its genetic material, not only does it inject its own, it's injecting some of that other type of DNA that's brought about from the old host. And this is how you can have genes that have been replicated or inserted into a new host. And this leads to what's known as recombination. It's something that we can't really do, but it's something that prokaryotes can do whenever you have foreign genetic material enter into the chromosome and add to it. So this adds a variability and a component of genetic variation for a usually asexual organism such as a bacteria. So that's generic transduction or general transduction. When we get to specialized transduction, okay, let's kind of go through this. We have attachment, penetration, great. We've integrated with a prophage. We have our prophage within our lysogen. When it comes to induction, where excision occurs, where it's normally cutting that prophage out, it's not very good at cutting a lot of the times. Sometimes it cuts a little bit too far. So whenever it cuts too far, not only does it take itself the prophage, but it's going to take part of the host with it. Like I said, this is a little bit more directed. And the reason that it matters more is because of the genetic layout of bacteria. Typically how their genes work is everything that is responsible for a certain process is in a row on its genome. So if it needs genes A, B, C, D, they're all within a row. So if it inserts right there, if it inserts and cuts, it's going to take all of those genes together. So it's going to accomplish a greater task. Versus in generalized transduction, if it cuts it up randomly, you just got to hope that you get all of those genes for that process. Randomly getting A, B, and C, D together into the next host. This is a little bit more directed. That kind of makes sense to everybody a little bit. Yeah, we'll get there. So here we're taking out, so see it cut a little bit too much. This is the host chromosome. And then it gets packaged up during maturation, and then it lyses out and it goes to infect a new one. And as you can see, whenever this new form of lysogeny occurs, it has the old host's DNA. Right? So we have a dark blue and a light blue. And that is showing specialized phage transduction. So you need to know the steps of specialized phage transduction. Specialized occurs in the lysogenic cycle, generalized occurs in the lytic cycle. What step comes after infection of a new host within the specialized phage transduction? I'll repeat that. What step comes after the infection of a new host after or during specialized phage transduction? Recombination. All right, that was prokaryotes. You want to talk about prophages for a second? Not prophages. Bacteriophages. Are bacteriophages good or bad?
Both.
Yeah, that's a good Gray Morale answer. It depends. That's the answer to most things. Yeah, it does depend. Okay. What about in terms of using them for human health? Do you think there's advantage to using bacteriophages?
Perhaps it's a
That's a good thought. Yeah, we do actually. So what you just kind of basically explained was genetic recombination. We do use that in genetic cloning experiments. Yeah. So yeah, we've taken advantage of it for biotechnology. Good. What else? Kind of gearing more towards health here in a way, but that is indirectly affecting human health. Yeah.
So since they only affect bacteria,
Very good. So say you have a bad bacteria. Say you have a salmonella infection. You want to get rid of it. What if we developed a prophage, and I told you prophages are very specific to kill just that bad bacteria? Why don't we do that?
Ethics?
We actually did. We used to do that in the late 1800s and early 1900s. And then antibiotics came along. Question? Emergency alert.
A bomb threat.
How close are we to the swish?
It's pretty close.
Yeah, in this case, we probably should.
Is it a bomb threat? I don't have my glasses
Give me
on,
a
so I
second.
can't read. Should we be concerned? Oh. Class canceled due to a bomb threat? That's crazy. Only in Texas, man. I thought Florida was bad. And we're known for school shootings. I'm gonna pause.